Okay. Here's what I consider to be the 5 steps. Attachment, phagocyte forms, phagosome matures, phagosome fuses with lysosome, ingested pathogen is killed. And those are the bullet points of what's happening. But we can go into a bit more detail than this and actually talk about what's happening in each of these steps. So that's what I want us to work on together for the next few minutes. Before we get to that, though, I just want to point out for this lesson 3 objectives. Describe this process of phagocytosis. So that's what we're going to work on right now. And then talk about how it contributes to immunity. That'll become a little bit more clear actually on Wednesday. We'll talk a little bit more about the steps of inflammation and how that contributes to the immune response. And then we're going to try towards the end here to take that general information, and this is the worksheet that I've handed out. We're going to talk about how do viruses, or in this case, how do bacteria actually overcome these defenses. Okay, we'll take a look at a couple of examples there. So let's talk about these 5 steps real quick. When we say that the first step here is attachment, what is that? What do we mean? What is attaching to what? Taylor, go ahead. Good. So usually, and that phagocyte is something like a neutrophil or a macrophage, are kind of our 2 biggest classes. Dendritic cells are specialized, are derived from the same white blood cell as macrophages, so we can include those as well. But that first step is they're attaching to something foreign. If we wanted to even more broadly define this, what we often say is that there's an attachment between a PAMP, PAMP, and a PRR. What do these acronyms mean? Pathogen-associated something something. Yeah. PAMP stands for pathogen-associated molecular pattern. So these would be things that we would commonly find on a large number of microorganisms. In other words, what do all gram-positive cells have on their outside? Peptidoglycan. That's a pathogen-associated molecular pattern. They also have teichoic acid. That's another PAMP. Gram-negative bacteria. What do they have on the outer membrane kind of sticking out? LPS. That's a PAMP. Okay? So PAMPs are things that are going to be common among a large group of microorganisms. Oftentimes we consider these to be foreign. They are not something that is naturally made inside of the host, so our immune system responds to it. PRR, on the other hand, stands for pattern recognition receptor. So these are the actual physical receptors on the surface of the cell, and this is just showing a couple of examples up here. These are those specific receptors that help to recognize foreign material. Okay. Some are located on the surface, and then sometimes we actually have some located inside of the cell. For cases when maybe a virus makes its way inside, then our macrophages and neutrophils might be able to recognize those molecular patterns inside as well. So that's the attachment. And the attachment here is just to make sure that the macrophage or the neutrophil, that phagocyte, grabs hold of the pathogen. And then the next step is the phagocyte forms, or the phagocytic vesicle forms. What's happening in that step? So what do we mean when we say that a phagocytic vesicle forms? I can rephrase this a slightly different way. How do we generally describe phagocytosis? What's another term or phrase that we might use to describe it? What was that? Cell eating, exactly. That's exactly what we mean with this phagocytic vesicle. So in this case, what we have is a couple of events. One, this binding encourages the macrophage to basically take its cell membrane and start to change the shape of it and envelop or wrap itself around that pathogen. And in doing so, it eats it and brings that to the inside of the cell. One of the ways that this can occur, if you see this image here, here's an example of a dendritic cell. And these are really long arms that are attaching to the bacterial cells. These arms are going to contract, and as they contract inward, you're going to start to see the membrane changes shape and wraps itself around the bacteria. And in doing so, brings it to the inside of the cell. Okay? So that's the actual, what we call the phagosome forming. Once that phagosome has formed, it begins to undergo maturation. What particularly changes as that phagosome matures? I'm not sure if it says it in here. Not necessarily. I'll give you a hint. Hydrogen ions are being pumped into the phagosome. What does that do to the phagosome? How does it change the environment? Drops the pH, exactly. So that acidic environment begins the killing process. Sometimes we refer to phagosomes as being somewhat like little bags of disinfectants. In this case, the acid serves as the disinfectant here. Okay? So the goal is to drop that pH to start disrupting proteins, start disrupting cellular structure. And then that phagosome fuses with a lysosome. And this is not to be confused with lysozyme, the enzyme. Okay? Lysosomes contain what? And I know a couple of people, Taylor, I know you had this kind of in your response, and I know somebody else did too. But what gets introduced into this phagolysosome, this joint product? You've got drop in pH. Taylor, do you want to share what you had? If you can remember. So the end result of this, yeah, exactly, is cell lysis. So when the phagosome and the lysosome fuse together, the lysosome is chock full of enzymes that actually don't become active until they come into contact with a low pH condition, which is what's in the phagosome. So these two fuse together, and what we have is activation of those enzymes. Many of them are proteases that their role is to break down the cell wall or other features inside the cell. We also find production of reactive oxygen species, hydrogen peroxide being the main one. We also find nitric oxide, some trace amounts of radical oxygen, which in any of these, the main goal of these is to essentially strip electrons away, in other words, oxidize the bacterial cell, leading to disinfection and killing of that microorganism. Okay. So here's where that event takes place, and eventually this will be exocytosed. We'll talk about this step as a critical step on Wednesday. Neutrophils, mainly all they're going to do is just get rid of this waste. Macrophages might actually repurpose some of it, and we'll talk about that on Wednesday. So those are the main steps of phagocytosis. A break in any of these steps can lead to the inability to degrade the microorganism. There are some individuals who are immunocompromised that actually lack the enzymes that produce these reactive oxygen species. And so because of that, we don't get the oxidative burst that occurs in the lysosome, and those individuals would actually be less able to control a bacterial infection. Questions on that process, or the steps that are involved in it? So this is what a macrophage or a neutrophil does once it's at a site of infection. The next question is, how does it even get there? How is it recruited to these sites of infection? And that is the inflammation response. So I want to take just a few minutes to walk through this together. And we're going to look at it from this large overview slide. And the other ones that I had up there are ones that more specifically get into specific steps of this. Usually the classical example shown in textbooks is somebody gets stabbed, and that stab causes some sort of infection. In this case here, somebody is getting stabbed with what looks like a broken piece of glass. That glass has microorganisms on it, and the bacteria get into below this basal membrane of cells here, so into the actual dermis. There are macrophages that are resident within this dermal layer. Okay? And pretty much every single organ has macrophages embedded into it to begin with. So in our skin, they're just below the epidermis. In our lungs, we have alveolar macrophages. In our liver, we have some, and sometimes these macrophages undergo a lot of different names that I blame the anatomy and physiologists for more than anything else. So Kupffer cells are macrophages. Those macrophages that are initially there, their goal is gobble, 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 eat up the pathogens. And what they will do then is when they recognize these pathogens, they respond. And one of those responses includes the secretion of two different types of molecules, cytokines and chemokines. And sometimes, and chemokines are just a specific type of cytokine. The function of these. Chemokines recruit. In other words, chemokines are chemoattractants. And you may remember this term from the beginning of the semester when we talked about bacterial movement. We talked about with bacteria how they will use flagella to move towards a chemoattractant and away from chemorepellents. We find that to be the case with eukaryotic cells as well. They will move toward or away from different chemicals. The chemoattractants, or the chemokines that are used here, are going to be used to recruit neutrophils and more macrophages to that site of infection. Okay. The cytokines are activating enzymes, or activating molecules. So rather than recruit, they are involved in taking a neutrophil and basically waking it up from a resting or a sleeping state. Some of these cytokines and chemokines, obviously the concentration is going to be highest where this infection is occurring or where those macrophages are releasing these from, but some are going to make their way into the bloodstream. And what we're going to find then is that this begins a recruitment of new cells to that site of infection. So let's take a look at a couple of these steps specifically. So the first thing we begin to find spacing between the endothelial cells that line the blood vessels. This is a process known as edema. Okay. That allows for for some of the plasma or blood plasma to flow into this site of infection. And blood plasma is actually rich in antimicrobial compounds, but also rich in compounds that help to enhance the immune response. Okay, so that's the first thing we find. Oh, and vasodilation. Sorry, the vasodilation is the separation or the dilation of the blood vessels, and then the separation of these cells from each other. The next things that we find are that neutrophils begin to they're kind of kind of bouncing along. I always think of tumbleweed. They're kind of bouncing through the blood vessels, and then all of a sudden they pick up a signal, or they pick up a chemoattractant. And their attitude changes. Does anybody have a bloodhound or a dog like a bloodhound? Or just a dog, period, that picks up on scents very easily? Dog picks up on a scent, and what happens? My dog starts pulling me in the direction of whatever he's smelling. And that's what these neutrophils do. And the way that my dog does it is he will kind of be walking, and all of a sudden he gets really low on all four and starts to move and sniff towards the direction of whatever he's trying to find. Neutrophils do a lot of the same things. They are kind of bounding around, and then all of a sudden they sense chemokine, and it's as if they stop, and they hold on, or they begin to hold on to the endothelial cells, or the cell wall, the walls of the blood vessels. Okay? This is a process known as margination. Holding on and starting to crawl instead of just bounding along, that crawling is the margination. And the goal here is that what those neutrophils are going to do is they continue to move towards higher and higher concentrations of chemicals. And then at some point, that concentration only gets higher as they move from the blood vessel out in between those vasodilated endothelial cells. That process of moving out of the bloodstream and into the tissue is known as diapedesis. The other term that the other figure refers to it as, and is sometimes used, is extravasation, but they mean essentially the same thing. So neutrophils are the first ones to respond. There's resident macrophages, but neutrophils are the first that make it to this location. And neutrophils are known for dividing incredibly rapidly. They are voracious eaters. Their goal is to just gobble up as much as they can and produce and break down those pathogens. Sometimes there are off effects of this. So, for example, you may experience with some wounds itching, additional burning, additional death of cells. Some of that death itself is actually produced by the reactive oxygen species and just the product of breaking down these pathogens. But what we always are hoping for here are at some point we've got clots beginning to form, and trapped in here would not only be macrophages and neutrophils, but we might also find bacterial cells, and eventually over time all of this plasma, pus, bacteria gets resolved. Okay. After neutrophils are recruited to that site, it takes about another hour or two for more macrophages to come. And we'll talk about their role then on Wednesday, because they play a role in transitioning the immune response from this, what we call innate immunity, to the adaptive immunity. Key point here with the innate immunity, again, it just recognizes patterns. It's recognizing for something that is foreign and common among many, many different types of bacteria or many types of viruses. All right. I'm going to stop there. I'm going to give you guys actually, let's say, I'm going to give you 2 minutes, or no, I'm going to give you about 3 or 4 minutes. And what I want you to do is to review with somebody else the 4 major terms here that are associated with inflammation. So term 1, margination, and these are in no particular order. Term 2, edema. Three, vasodilation. And then last, diapedesis. And I'd like you to do two things. One, put those terms in order in terms of when they happen in the process of inflammation or when we first see them. And then secondly, a term or two to describe or help to define those steps for yourself. Let's go till about 11:35, and then we'll finish up.
Can I ask you about allergies?
Yeah.
So, histamine that's produced.
Yeah. Any questions on those steps, terms? Okay. We'll leave that as is for now then. But where I do want to go then is to talk about some examples in which pathogens will respond to this. The first one actually is something that we've seen in lab. So Streptococcus pyogenes. No, we haven't seen this one in lab. Sorry, I'm thinking of something completely different. Some organisms will evade the inflammation response. For example, Streptococcus pyogenes produces an enzyme known as streptokinase. This enzyme breaks down blood clots. Okay? So the streptokinase enzyme breaks down blood clots. The benefit of that is it allows the organism to escape these localized regions where infection is occurring, to escape the fibrin clot that has formed, and it actually allows for the organism to spread its infection. So what we find with Streptococcus pyogenes, when untreated, it makes its way into the bloodstream and then can cause infection. If you've had strep throat, nowadays we can treat strep throat pretty darn easily with antibiotics. But if you were an unlucky individual to have strep throat in, say, the 1930s or before, one of the common associations with strep throat was scarlet fever. And scarlet fever is a rash that's produced, which is basically a rash is inflammation produced by Streptococcus pyogenes and some additional toxins it produces to make their way out of these clots that would form around them and into the bloodstream. Okay. We actually see other ways in which organisms can avoid the immune response. And we're going to take a look at a couple of examples related to macrophages and phagocytosis. Here's our first one. An enzyme we've talked about before, at least in lab, coagulase. Does anybody remember what the coagulase test is? Taylor? Not so much antibodies, but we use plasma for this test. Clumping. We're looking for clumping. Yeah, there's some similarity there because, like with an antibody test, we look for clumping as well. This one, what we're looking for is clumping or clotting to actually occur. So what Staphylococcus aureus does is if it contains the enzyme coagulase, it actually will form a clot around it. All right. What effect is this going to have? And so that's what we're going to take a look here. So just to kind of orient you with this image real quick, we're only going to focus on the one in red here in the wild type. The red here is delta COA. In other words, this organism has lost the gene that encodes for coagulase. So the wild type, you can just put a plus coagulase next to it. The delta COA, you can put minus coagulase next to it. Given that, what I want you to do first is just describe for me the results in figure A here. So this is looking at survival of Staphylococcus aureus, either the wild type or delta COA, in blood. So how much is there in blood versus how much they began with. Okay? So just take a look at that figure, describe it. What's happening in this figure? Or what do we observe? In which population, Allison?
In the ones without coagulase.
That's a great explanation, or that's a great description. Decrease in survival with those not having coagulase. What would our interpretation of this be? There's a couple of possible ones based on just this piece of evidence alone. But, Allison, what would you interpret this to mean? Exactly. Coagulase protects. Not only does it protect, but take a look at the graph here on the right. This one, they took mice. They infected them with wild-type Staph aureus or Staph aureus lacking that COA. So just again, focus on the black and the red. Those infected with Staph aureus, all of them are dead within 24 hours after infection with the amount that they inoculated with. When you knock out coagulase, it took 3 days to kill them. In other words, with the addition of coagulase, not only does it protect the organism, Staph aureus, but it makes it more virulent or more pathogenic. Okay? It allows for Staph aureus in those cases to cause illness and to kill at a much faster rate. And then the question becomes, why? What is the benefit of forming a blood clot here? I'm not sure if I have this. Well, is this it? No. The answer comes down to the following. I'm going to draw a picture real quick, and then you can, we'll see if you can interpret this. Oops. So let's make these our wild type, and then our minus coagulase. And let's take a look at percent in let's look at the percent of these that we would find in macrophages or associated with macrophages. Oops. So wild type cells, we might only find like 25% of them associated with macrophages. Those without coagulase, let's say it's 100%. So without coagulase, we find more often that Staphylococcus aureus, if they lack this coagulase gene, they're more often in contact with macrophages. So what is the benefit of coagulase then? If we were to interpret that, what is the benefit of coagulase?
Does it benefit them to be more in contact with macrophages because macrophages recognize?
So this would actually, so being in contact, since macrophages recognize them, would actually be a downside, right? But you're getting at a good point. Look, Taylor, go ahead.
The clot kind of keeps the bacteria like centralized to one location, so the macrophages can just there. Whereas if it's like widespread throughout the body, the macrophages have to go to many different places, and then it's less concentrated.
But wouldn't that be a good thing for the host?
No, it would probably be worse because if it spreads throughout the blood.
Okay, I see what you're saying. I've got your argument now. I'm going to draw something a little bit differently. Purple, that's our Staph aureus. Okay. Here's its clot. Purple tells us what about its gram status? Positive. What's true about gram positives? What? Thick layer of peptidoglycan, which in the case of immunology is what? we would classify it as a PAMP, pathogen-associated molecular pattern. That's what gets recognized. Now, a clot, on the other hand, the clotting is of fibrin or fibrinogen. That's us. Does our immune system respond to itself? If we have healthy immune systems, it shouldn't, right? A healthy immune system is one that responds to something that's foreign, not something that's self. So what's the coagulase doing here? Yeah. Harry Potter fan. What's it doing? Think of something Harry Potter-ish, maybe the first movie. What does he receive for Christmas? Or the first book? What was that? An invisibility cloak. How is this like an invisibility cloak? Shields them. It prevents them from identification. Okay? So what this coagulase is doing here is by cloaking itself with something that is the host, it's actually preventing the recognition of those pathogen-associated molecular patterns, and therefore, it's preventing the phagocytosis from occurring. So if we go back to this real quick, the reason why so many are surviving in the blood is because they're cloaked, they're hidden. Whereas those without coagulase, their molecular patterns can be detected. Okay. Annie?
Is this clot different than the clot that the host forms?
There might be some differences in it. I suppose it would be more quote unquote clean than the clot that you find during.
Isn't that clot that we form?
The material is, yes, it is, but the reason why, you're absolutely right, it is beneficial because the clot helps to prevent the spread of infection, and also embedded in that clot already are things like macrophages and neutrophils. So here is an example where actually clotting can be a detriment to the immune response. Let me give you one more example that kind of follows the same path, and we'll leave it. There's a third one that I want to talk about, but I think we'll save that for Wednesday. You may remember when we talked about horizontal gene transfer, this experiment where Griffith takes rough and smooth Streptococcus pneumoniae, and he exposes mice to them, and those with the rough phenotype, son of a gun. So those with the rough phenotype don't kill the mouse, and those with the smooth phenotype do kill the mouse. Does anybody remember what the difference between the rough and the smooth-looking ones were? Some bacteria have a structure that makes it really hard for the macrophages to detect them. A capsule, or a slime layer. A glycocalyx is another term that we use for these. And those organisms that have a capsule, that thick sugar coat actually prevents the identification of the pathogen-associated molecular patterns. In some ways, the clot formed by coagulase and this capsule, they serve the same function to protect the identification of the microorganism by the immune system. So this example here is just showing you an experiment, smooth versus rough, inoculated into mice, and then I can't remember if it's like 10 hours later or what, they measure, well, how much is still there. The smooth have increased in numbers, the rough have remained the same. And the inability to increase in numbers is because those rough, and not having a capsule or ability to form a capsule, they're being recognized by macrophages more frequently, being broken down and killed. Okay? So that's just two examples that we've seen that can be avoided. I'm just looking at this last one here. We'll take a look at Mycobacterium briefly on Wednesday if I feel we're up for it. But otherwise, pay attention to those two. You may see a question like that on the upcoming exam, which is 3.5 weeks out. All right. See you in lab tomorrow. Have a good afternoon.